Hey guys, welcome back to Jeevashastra Abhyasa. I'm your educator Niranjan Hegde. So in the last video we have completed the microbes in the human welfare. So if you haven't watched the whole topic, I'll put up in this YouTube icon. You just go and check whole the playlist. Today in this video we are going to begin a new chapter which is known as biotechnology and its application. So first we have to know about biotechnology and then we'll move to the application. So we'll just know what is the definition of biotechnology and we'll come back to its applications. So what's the wait? Let's begin. Biotechnology can be defined or it can be easily defined as when by splitting up of the whole term itself. Biotechnology. Okay, so if we split it, we are going to get bio plus technology. What is this? Bio means life. Okay, that is life. Technology itself, the technology, that's nothing. So nothing it. So the name itself. So using the technology, if we alter the life means alter the organism for the human benefit. That particular thing is known as biotechnology. Simple, very, for human benefit. If we exploit the organism's life in a healthier way, remember that, in a healthier way, in an ethical way, so then we can call it as biotechnology. Okay, so now this biotechnology, so altering the life, so altering the different varieties of species which will give us very different benefits, so that is known as biotechnology and its applications. Okay, so what are the different types of applications we can see with the help of this biotechnology? So the mainly so this biotechnology can be applied in the agriculture field. First thing, it is agriculture field. Agriculture. The second, we can apply this in medicine. And third one, we can use the biotechnology for producing transgenic animals. And these are the three applications what we are going to study in this particular chapter. And at last in this chapter, we are going to study about the ethical issues. So what is ethical issues? All those things which is related to this particular biotechnology that we are going to study. Now one thing, so this three is a little bit briefer. So I'll try to cover these two today and this two in the next video. So tomorrow we'll finish up this particular thing by tomorrow evening or on Sunday morning itself. So on these 3 days we are going to finish it off. So let's see how biotechnology is applied in agriculture and medicinal field. So let's study it. Okay, so I'll rub it and we'll start it. Okay, this is how this particular biotechnology has applied over agriculture. So how it is applied is, so in the 1960s, the green revolution started all over the world. Green revolution is nothing but the production or the thinking out of the box in the field of agriculture. So in that particular, now also it is going on. So we can say that green revolution is where new technology of agriculture has been developed. With that, new varieties of plants are also developed. So how then it is developed? Using the biotechnology. So, new varieties of GMOs GMOs has been developed at that time. GMOs is nothing but genetically modified organisms. What is this? This can be asked for the abbreviation. Genetically modified organisms. Okay, it is known as genetically modified organisms. So these genetically modified organisms, one of them is the GM plants. That is genetically modified plants. So it has been created using different types of bacteria. Different kinds of bacteria were responsible for the modified plants, genetically modified plants. That is bacteria. So we have studied it before also in the last video even. So just try to remember it. And next point is, so how these GM plants were or in what objectives when they kept some objective they have kept for the development of GM plants, right? So there are some objectives have been achieved by creating a genetically modified plants, which we are going to discuss. The first thing, the Green Revolution took place because there were a lot of floodings were taking place all over the world. So the scientists thought that so the bacteria, the organisms which help them to tolerate the very crucial times of period of time, that is stress, we can go abiotic stress. If the plant can survive the abiotic stress, for example flood, one of the example is flood, then a drought, then cold, then hot. So these type of weather conditions, if they survive, so that is abiotic stress. If they have developed this kind of they have achieved this objective by using the genetically modified organisms. So for example, we have studied in the last, that is Bacillus thuringiensis, BT, right? Remember that, Bacillus thuringiensis. Next, they have developed a type of plant which is going to give yield, giving yield in a shorter period of time. In a shorter period of time. So the paddies were at that time 6 months it was took. Now it has been reduced to 3 to 4 months crops, right? So we can cultivate 3 times. So that type of inventions has been done, because if it is not done, so the food shortage has been started at the same time itself. Now this particular thing is interconnected. Next point what I am going to discuss, that is, so as the month as the yield period has decreased, so there were the decrease in the yield loss also started. Decrease in the yield loss because of the total number of days the plant would survive. So that is what this one also interrelated, right? Because the yield loss has came down to the point. So the next one is it has been also the yield what we are getting is also has been nutritionized, or the increase in the nutrition has been made in this plant using the biotechnology. Increase in the nutrition of the yield. Such type of genes have been introduced into the plant so that they have enriched the yield quality, right? So that was very important. Then also the mineral content of the plant, remember, of the plant has also been increased so that they can survive the different kinds of conditions provided by the environment. So this was possible because of the different kinds of bacteria. Now for your study, one example has been given. That is, I'll just rub it out. So that is Bacillus thuringiensis. Okay, directly if I come to the example, that is the example was given was BT cotton. This is the direct example what for you in the agriculture has been given. One of the example. Okay, BT cotton. What is BT? That is Bacillus thuringiensis. 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 Okay, thuringiensis. Thuringiensis. So this bacteria's a particular gene has been inoculated into the cotton plant. So why? Because for the purpose of pest resistant, to make them as pest resistant. Okay, remember that, to make them as pest resistant. That is also how the scientists observed, or the agriculturists observed, that Bacillus thuringiensis releases a toxin a toxin a toxin which is highly toxic for this particular pests like insects, bugs, then borers. So for them this is particularly very toxic. Then which particular gene expresses, they found out this, which is going to express this toxic material, right? So that was cry. The gene was named as cry. The cry was the name of the gene which expresses the toxic content in the Bacillus thuringiensis. For example, so this cry has been inoculated into the cotton plant. Okay, so what is that? There is cry 1AC. So this was the gene which has been inoculated into the cotton plant. There are some examples are also there. Cry 2B. Cry 2B is also one of Cry 2AB is one of the example for the plant, this type of plant, which is again cotton bollworms. So when bollworms attacks the cotton plants, so that in that area this particular gene is inoculated into the cotton plant. So it proved a very good yield for the farmer. It turned out this type of plant, and it is successfully now also it is running. So everywhere BT cotton is very famous, and it produces a good quality cotton also. So this is a one example, so which we have discussed. Now let's move to the next part. So but you have a question in your mind. So what is this toxin in the Bacillus thuringiensis? It's a protein part. Okay, it's a simple protein part which is toxic to the pests. Okay, simple that. Okay, so next we will move to the some of the pest resistant plant which has been done through the introducing the RNAs into them. So let's learn that. So now we will discuss about the second example, that is a pest resistance plants. For example, we are going to take the plant of tobacco. Okay, tobacco plant we are going to take, and we are going to discuss the main thing of how these tobacco plants get affected with a particular nematodes known as Meloidigene incognitia. This was the nematode which affects the roots of the tobacco, and it ultimately decreases the yield of the plant. Okay, fine. So now what is and how we will discuss. First, let's directly move to how it is going to be stopped with using the biotechnology. So we are going to use the agrobacterium. So we are going to use this particular agrobacterium. So where the DNA the DNA will be present inside it. So then why we are going to inoculate that particular agrobacterium into the roots of the tobacco? One very important thing. So what happens when it the Meloidigene incognitia enters into the roots of the particular tobacco plant? So there will be a stoppage in the mRNA formation because of slicing of the mRNA takes place. The slicing of the mRNA takes place due to the blockage. This particular nematode blocks the mRNA formation. So that is slicing takes place, which results in the form. There will be no formation of protein. That means there will be no formation of the antibiotics is going to happen, right? So that is what happens when agrobacterium is introduced into that. So there will be a DNA is also going to be induced. So which produces the RNA. So here in this RNA there will be antisense and sense strands, sense RNA strands will be generated, which ultimately give rise to the double-stranded RNAs. So there will be a double-stranded RNAs will be generated, newly formed. So then it directly forms the antibiotics in the host, and there will be the effects of this particular nematodes will be decreased. So this is how we can use the biotechnological advancements for the decreasing the diseases in the plants and other animals even. So this is what the application in the agricultural field. So now we will move to the next one, that is in the medicinal field. Okay, so in the medicine field, so how the biotechnology is applied. So hinting, yes. So here mainly the recombinant DNA technology, when it is being introduced, so this particular changed the way of forming a medicine. Okay, so then why and how? So here, so this particular recombinant DNA technology introduced the formation of different varieties of bacteria and the fungi and the cyanobacteria for the production of different varieties of enzymes which are required for the human body if the human body is deficient. So this is also like the catalyst. Here they become a catalyst, like a catalyst. They are not exactly the catalyst, so they will become a catalyst. So that is how we are using the organisms in the biotechnology for the human benefit, right? So in the medicinal field, the thing happened is that there are a lot of enzymes are required for our body. So we are lacking some of them. So then we find the deficient in our body, which will turn definitely into the diseases, some of the diseases. For example, the one example is the insulin. The insulin is produced in our body by the pancreas. So how it is produced, first let's learn, then how it has been, biotechnology has been implied, I'll tell you. The insulin is produced in the pancreas, as you know that. So there will be a pro-insulin, okay, so which is entangled with the three types of peptide bonds, the three types of peptides. One is peptide A, peptide B, and peptide C. So peptide B, peptide A, and peptide C, all they are together. Okay, so then that pro-insulin, the pro-insulin is going to get matured. So when the pro-insulin matures, when it starts releasing, the pancreas starts releasing, that will be time of maturation of pro-insulin into the insulin. So then at that time what happens, the insulin is formed. How? So only by the two peptides, that is A and B peptide, with a disulfide bond. So you can see over here by the disulfide bond. So the free C peptide will be freed. So it's like an enzyme. So just think it is like an enzyme. So this part will be released. So the lack of insulin causes the diabetes mellitus. You know that, right? So this is how insulin is formed, and insulin deficiency causes what? Diabetes, right? Diabetes. Diabetes will be caused. Okay, fine, fine. So then how to solve this diabetes? So the scientist has easily available insulin was through the pig, through the cow, through the goat, all those things insulin was available because they are also having the pancreas. So insulin was taken out from them and it is being introduced into the human body, extracted. So then what happens? Some in lot of patients there will be a lot of complications started taking place. Why? So they started finding it, why it is happening like that. So then they found out that in those pigs, cows, and the goats, this particular C peptide was missing. The C peptide was missing. So that will directly the insulin was produced. So then what we can do? How we can do? Now what they did, the scientists did that using the recombinant DNA technology, they inserted the proinsulin directly. They inserted the proinsulin. They extracted the proinsulin from the pancreas and they introduced them into the. So the person was having the there was no insulin is formed. Proinsulin was there, but insulin was not getting formed. So that's why what they did, the scientists, they introduced the proinsulin using the recombinant DNA technology. That particular gene identified them and which produces the proinsulin. They introduced them into the E. coli bacteria, that is Escherichia coli bacteria. They introduced it in this particular type. They inoculated into the E. coli and they supplied a particular type of environment like a pancreas. They introduced the nutrition to them, and that very importantly, it has given us the A peptide and the insulin directly. So that the same procedure took place in the E. coli, so which triggered the proinsulin has converted into the insulin, and thus that insulin is multiplied several times, okay, using the same technology. And before that, after formation of this particular thing, they also introduced that particular gene which directly found this one, and they took that insulin and now it has been administered to the individuals who are suffering from the diabetes mellitus. So this is one of the example for insulin. We will discuss more type of medicinal application in the next video. So be there until the next video. So let's end the video here itself because it will be a lengthy video, so you will lose the interest to watch it. So thank you for watching. Like and share. Don't remember? No, do remember to share and like. Okay, fine. Until next time, let's grow together. Jai